Hello everyone and welcome back to my biochemistry series. This is going to be the fourth installment of lecture 2. If you remember, lecture 2 is about the cell and subcellular organelles. And in this part, we shall focus on the nucleus. We shall study the nuclear membrane, nuclear pores, nucleoplasm, the nucleolus and DNA, which is the genetic material of eukaryotic cells. So in this slide, you can see a label diagram of the nucleus. I suggest that you spend a few moments and have a good look at it. You can pause the video at this point if you want to. The nucleus is the largest organelle in animal cells, and it is enclosed by a double-layered membrane which is continuous with the membrane of the endoplasmic reticulum. The nucleus controls and coordinates various activities of the cell, such as protein synthesis, cell division, growth, and metabolism. The nuclear membrane is not continuous in its entirety. It has several thousand openings which are called nuclear pores, which connect the inside of the nucleus to the cytoplasm. And at the edges of these pores are attached large complexes of protein molecules, which are called nuclear pore complexes or NPCs for short. And these complexes are typically around 90 to 100 nanometers in diameter. However, the central open lumen of each pore is only around 9 nanometers in diameter, and through this 9 nanometer wide channel, molecules with molecular weight of around 44,000 daltons can pass through with ease. Now we'll talk about some of the molecules which actually move into and out of the nucleus through these nuclear pores. So molecules moving out of the nucleus are RNA and ribosomal proteins. And molecules moving into the nucleus are DNA polymerases, carbohydrates, signaling molecules, and lipids. Moving on to the nucleolus, it is a dense non-membranous mass that is closely associated with the inner nuclear membrane. And the nucleolus is where the transcription and assembly of ribosomal RNA occurs. Then we have the nucleoplasm, which is the ground substance of the nucleus, just as the cytoplasm is the ground substance of the cell, and the nucleoplasm contains enzymes such as DNA polymerases and RNA polymerases. An important piece of information that you must know is that not all eukaryotic cells have nuclei. For example, human RBCs lack nuclei, and this is basically an adaptation. The absence of nuclei in RBCs reduces their oxygen consumption, their oxygen demand, and because of this they are able to deliver more oxygen to the target tissues. And since they lack nuclei, RBCs also lack genetic material and they do not replicate. Now let's move on to DNA, which is the genetic material of living organisms and is found inside the nucleus. In eukaryotes in particular, the DNA does not lie freely. It is bound to histones, which are basic proteins, and the ratio of the DNA to histones is roughly 1:1 by weight. A little bit more about the histones. These are of 5 classes: H1, H2A, H2B, H3 and H4. Now let's talk about how DNA and the histones are actually bound to each other. In reality, DNA and the histones are organized into bead-like structures called nucleosomes. So a nucleosome consists of one histone octamer, which has two molecules each of H2A, H2B, H3 and H4 histones, and around each octamer are bound roughly 147 base pairs of DNA in nearly 2 turns. And in between two adjacent nucleosomes, you have some linker DNA which is bound to one H1 histone molecule. So together the nucleosomes and the linker DNA form what is called the beads on a string model of eukaryotic DNA. The nucleosomes resemble the beads, and the linker DNA, which is bound to one H1 histone molecule and which is more or less linear, resembles the string. So the beads on a string structure is around 10 nanometer wide, and it can be stacked even more tightly upon itself to form what is called the 30 nanometer chromatin fiber, which has a width of 30 nanometer. And during cell division, this 30 nanometer fiber gets stacked upon itself even more tightly to form chromosomes. So chromosomes are a highly condensed form of chromatin fiber which appear during cell division. And a single human chromosome has around 1 million nucleosomes. So here is a series of diagrams that show the progressive stacking of genetic material, that is DNA, inside the eukaryotic cells. So first of all, here you have the classic beads on a string model, which is around 10 nanometers in width. And then the nucleosomes can be stacked upon themselves to form the 30 nanometer wide fiber, which is further stacked upon itself even more tightly to form chromosomes during cell division. Now let's talk about why this stacking is necessary. It is necessary because if we unstack all of the genetic material, all of the DNA inside a cell, inside a single cell, it is around 2 meters in length. And in contrast to that, the nucleus of the cells where all of this genetic material is present is of course microscopic in nature. So stacking is necessary to shorten the length of the DNA and fit it inside the cell nucleus. Now we shall discuss a clinically important topic related to the nucleus. Specifically, it's about a disease called the Hutchinson-Gilford progeria syndrome, HGPS for short. And HGPS is also known as progeria, which means premature old age, or simply Hutchinson-Gilford syndrome or childhood progeria. So what happens with HGPS is that it is due to a genetic mutation. So there is a gene called the LMNA gene, which codes for a protein, a nuclear protein called lamin A. So mutation in the LMNA gene leads to a defective or abnormal form of the protein lamin A, and this abnormal form is called progerin. So due to the abnormal nuclear protein progerin, there is accelerated aging in HGPS soon after birth. Fortunately, the incidence of this disease is very rare. It is around 1 in 5 million births. Typical features of HGPS are a small face relative to the head size, loss of hair, wrinkled skin, etc. Unfortunately, this disease is invariably fatal, and death of the affected individuals occurs in the teenage due to cardiovascular complications like myocardial infarction or stroke. Here you can see a generic and very archetypal image of a child with HGPS. You can see some of the typical features such as the large head and the face, which is small relative to the size of the head, the hair loss, the wrinkled skin, and also some other features. To sum up, in this lecture about the nucleus, we have studied the nuclear membrane, nuclear pores, the nucleolus, nucleoplasm, and DNA. And we have also learnt about Hutchinson-Gilford progeria syndrome, which is caused due to a defective nuclear protein. So that's going to be all for today. Please stay tuned for the next lecture.